next talk is by Ruth Hirschberg. Oh, no, sorry, by Ian Kutch. Sorry. That's
all right. All right, thank you. Can you guys hear me? Yeah, my name's Ian Kutch, and I'm a PhD student just up the road at the University of Central Florida. And I'm going to talk a little bit about some of my dissertation work on the Y chromosome and its potential role in the evolution of autosomally coded traits. And so from my title, you can guess that I'm interested in sex chromosomes, and particularly I'm interested in the heterogametic sex chromosomes. The reasons why people might be interested in this chromosome are many. My interests lie in that it's a chromosome that exists only in one sex. And given that sexual conflict is pervasive throughout the animal kingdom and pervasive across the genome, this chromosome becomes a really interesting place for genes to maybe be sheltered from sexual conflict. However, there are many other aspects of Y chromosomes that make it a little less interesting when it comes to evolutionary studies. Particularly in heteromorphic sex chromosomes, we see that Y chromosomes typically have low gene content, very few numbers of genes relative to the X chromosome or autosomes. The genes that it does contain have really low levels of nucleotide variation, and they typically are comprised of mostly heterochromatin. And so all of these kind of characteristics come together to make this kind of an uninteresting chromosome from an evolutionary perspective. However, more recently, we have some new insight into the Y chromosome, showing that Y chromosomes are actually influencing the expression of genes throughout the genome coded for on the autosomes and X chromosomes. This is nice work done by Bernardo Lemos and his colleagues, and they show very nicely that putting different Y chromosomes into the same genetic background dramatically affects the expression of many genes. And so this little figure here, we got Y chromosomes from different countries and different localities put into the same genetic background, and we see drastic levels of gene expression differences across all of these candidate genes. I put a little subset up there of immune genes, and I'll talk about that a little bit later. But this is not just happening in immune genes. This is happening in hundreds of genes throughout the genome. And the mechanism that is gaining the most support, I believe, is the idea that the Y chromosome is kind of acting as a heterochromatin sink. So these Y chromosomes are variable in the amount of heterochromatin they have, and that variation has downstream effects on the chromatin dynamics of the rest of the genome. And this can lead to those differences in gene expression. So this effect is called a Y-linked regulatory variation, and I'll talk about it for the rest of my talk. I'll call it YRV. So this kind of new function of the Y chromosome, newly found function of the Y chromosome, is very interesting, and it's led us to kind of revisit the Y chromosome for some hypotheses, specifically related to sexual dimorphism. So sexual dimorphism is when males and females have different versions or different levels, different trait levels for the same trait. And it's very interesting because given that males and females share the same genomes, there should be a large constraint to the evolution of these sexually dimorphic traits. And given that they are sharing the same genome, we know that these differences must come about from the differential expression of the same sets of genes in males and females. And so that's sexually dimorphic gene expression leading to those sexually dimorphic traits. And the hypothesis that we want to address is, can YRV, this Y-linked regulatory effect that I just talked about, influence the evolution of sexually dimorphic gene expression? And we think it may do this by providing sex-specific genetic variation, or sex-specific variation in gene regulation that selection may be able to shape, giving us these sexually dimorphic traits. So what we're saying is that YRV may facilitate the adaptive evolution of sexually dimorphic gene expression. We know a little bit about how sexually dimorphic gene expression is achieved in many different animals. I'll go through a little bit of that right now. We know that in all metazoans, the double-sex MOB3 related transcription factor family initiates gonad development, and we know that sex-specific gene regulation is what leads to sexual dimorphism. In vertebrates, these gonads lead to sex-specific hormones, which can directly differentially regulate the genome. But in invertebrates, they don't have those sex-specific hormones, so they're likely using spliceal variants of double-sex or MOB3 related transcription factors to directly differentially regulate the genome. What we're proposing is that the Y chromosome or W chromosomes, heterogametic sex chromosomes, can directly influence the regulation of genes leading to sexual dimorphism. Now, in order for YRV to facilitate the evolution of sexually dimorphic gene expression, it must exist within natural populations. It must influence fitness-related traits that are shared by both sexes. That's an added little part there. And it must be comprised, at least in part, by added genetic variance. And really, this is just putting this variation in the context where selection works. Selection works within populations, on fitness-related traits, and on additive genetic variation. And so we wanted to kind of test these predictions here to support this hypothesis that YRV may be playing a role in sexually dimorphic gene expression. To do this, we took fruit flies, and we went to a local population of fruit flies, collected wild fruit flies, and we got 30 different Y chromosomes from this population in Orlando, Florida. We put the 30 different Y chromosomes into an isogenic background, very similar to the methods of Bernardo Lemos. We call these Y lines. The only thing differing between these lines should be the Y chromosomes. And we tested for a couple candidate genes for differences in gene expression across these Y lines. We picked immune genes because that's what my lab works on, immune function, and Bernardo Lemos' work showed a nice effect on immune function genes. And so we used qPCR to see if there was variation across these Y lines. And what we found were that in females, there was no significant difference across those Y lines, but in males, we had significant differences in gene expression in 2 of the 3 genes. These females worked as a nice negative control across the Y lines. They are isogenic, so we didn't expect to see any variation, and we didn't. In the males, we do see significant variation, suggesting that these Y chromosomes from a single population are influencing the expression of these immune genes. To take this from within population variation on immune gene expression and start to look at a fitness consequence, we tested for immune response in these organisms. So we took, again, 30 different Y lines from the same population. This time we infected them with a gram-negative bacteria, Serratia marcescens, at a very consistent LD90, and we looked for variation in their time to death using these Drosophil activity monitors. So kind of a proxy for survival against this pathogen. We used a proportional hazards model to look for differences across these Y lines, and again, in females we see no differences, but in males we see significant differences. Again, the only difference in those males being the Y chromosomes that they were paired with. What we could also do was take this data and kind of compare it to the gene expression data that we had from the previous study, as 19 of our lines overlapped in both of these studies. And we had this nice correlation between levels of immune gene expression and the time to death of these organisms, showing that lines that had high levels of immune gene expression from Y chromosome variation lived longer, and lines that had low levels of expression died earlier. So that was a nice little correlation that we found. The next thing we wanted to do was get at Y-linked additive effects. Is there additive genetic variation for this trait that selection can actually shape? And some other studies have looked at this, but within the context of sexual dimorphism, they had a few caveats. Some of the studies looked at a male-specific trait, so we're not going to get the evolution of sexual dimorphism of that. And other studies paired non-co-evolving genetic elements, and so we're not talking about within population variation. And so we want to address that by using co-evolving Y chromosomes and genetic backgrounds, and using a male and female shared trait immune function. And so we took our population of Y lines, and we took 4 of those Y chromosomes, and we crossed them with 4 different genetic backgrounds, 4 different autosomal haplotypes. So a little matrix design there, 4 different Y chromosomes, 4 different backgrounds, cross them together. And what we were able to do was look again at response to this immune challenge, looking at time to death. And we could kind of parse out these model components to tell us a little bit about the additive effects in the system. We could look at variation that compounds within a background. That would be background effects. We could look at variation that compounds within a Y chromosome across backgrounds. That's our Y chromosome effect. That's the additive component. Or we could look at the interaction between Y chromosomes and backgrounds, the epistatic component. And so here's the results from that study. We see significant background effects, which is good. That means our backgrounds were variable and they contributed to variation in survival of these organisms. We found no significant Y chromosome effect, the additive effect, but we see a significant influence of Y by background interactions. So this is a lot of, this is the epistatic effect. What we were able to do then was take our little cross of 4 Y chromosomes and 4 backgrounds, not really a population, but of this study system, we could parse out the variance components attributed to these different model components. And what we see is that the Y by background interaction, that Y-linked epistatic effect, was making up a lot of the genetic variance for this system. So 43% of that genetic variation was attributed to the Y by background influence. That's a pretty large magnitude. And so we found very little evidence for additive effects, but significant epistatic effects. So ultimately, we want to draw some conclusions from this. I proposed the hypothesis that the Y chromosome, or YRV, could influence the evolution of sexually dimorphic gene expression. We did find that the variation exists within populations and influences some components that influence fitness, but we find no evidence of the additive effects that would translate to additive genetic variation on the population level. There's some caveats there. This is one trait. These were only 4 Y chromosomes and 4 backgrounds. There may be additive genetic variation out there, but our design was pretty robust to look for additive effects. And seeing that we saw none in this study, it leads me to think that YRV additive effects, if they're out there, are probably minimal for this trait. There's another component, though. We found these epistatic effects, and how the epistatic effects on the physiological level, which we were estimating, translate to epistatic genetic variance components and additive genetic variance components on the population level. It gets a little interesting, and we can't really parse that out with our 4 Y chromosomes and our 4 backgrounds. Genetic variance components depend on allele frequencies and magnitudes and directions of the interactions, and so if we really want to understand how epistasis is influencing the population level genetic variance components, we need to not look at lines, we need to look at populations, and so that's something that we're doing in the lab. So we don't really know exactly how that's going to parse out. It kind of depends on the population. However, the epistasis that we found complete rank order changes in the Y chromosome effects, given the background that they're in, and of this large magnitude, we think that YRV can have significant implications for monomorphic selection. And so essentially this epistatic YRV is putting males and females working in different genetic architectures. And so females are dealing with their autosomal and X-linked components of additive effects and interactive effects, but males now have this added component of the Y-linked genetic variance. And this, if it's largely epistatic, can really have some consequences for monomorphic selection. A lot of epistasis of the magnitude and direction that we've seen could significantly increase phenotypic variance, and it could reduce the heritabilities for traits while they are in males. And so we have this potential constraint for YRV, for Y-linked epistasis. So ultimately, to really understand what this means for the response to selection, we need to move away from lines, and we need to start looking at populations. So we're starting to do some experiments on populations of varying allele frequencies, of Y chromosome frequencies, and seeing the consequences of that. Or we need to start looking at natural estimates of YRV, and hopefully we can get to that soon. And with that, I'd like to thank the organizers of this group, very excited about it, and my advisor, Ken Fedorka, and our lab members.
Have time for a few questions?
When you are looking at the correlation between the expression of those antimicrobial peptide genes and the time to death in the Serratia infection, presumably that expression is measured in uninfected flies, so that's the constitutive level of expression.
Yeah.
Do you know anything about inducibility of immune response, and do you have any hypothesis for why you've got this different basal level of expression?
Yeah, so the previous work on the Y chromosomes effect on gene expression was done on baseline levels, not induced levels, and so we followed that kind of system. And then when we did the infection and just ran the correlation, it was nice. And so what it looks like is having a higher baseline expression level, at least from a Y-link perspective, correlates with your ability to fight the infection. So no, we did not do the induced measurement, but that would be a nice thing to look at. Yes?
I was wondering how this system would translate into a species that had autosomal recombination in both sexes, whereas in Drosophila, the males, like the autosomes, remain constant in the
Yeah,
population?
yeah. We utilize that in our breeding design to get Y chromosomes into different backgrounds. The Y chromosomes would still influence the autosomes differently, even if they had recombination in other systems. Or at least that's what we think.
Thank you.